Okay, let's get the show on the road. Welcome to the Trobe's Research Infrastructure and Services Seminar for New Researchers.
I will begin with the acknowledgement to country. And so I'd like to acknowledge that this event and our participants are located on the lands of many traditional custodians. We celebrate the diversity of Aboriginal peoples and recognise their ongoing connection to the land and waters and value their unique contribution to the wider Australian society. We pay our respects to Indigenous elders, past, present, and emerging, and extend this respect to any Indigenous participants joining us today. So, welcome to you all. My name is Christopher Adder. I'm the Director of Research Infrastructure at La Trobe. And if I were to describe my role in a nutshell, it's all about supporting the conduct of research across the university and enabling and accelerating new research outcomes. And so the portfolio of the Office of Research in Infrastructure encompasses looking after our research equipment and facilities, including our research platforms, research infrastructure capital funding scheme, and also other large equipment schemes such as the LEAF scheme, and also digital research is in there as well. So today you hear from a number of my colleagues across the research platforms, and also Nick, who's going to talk about digital research. Now, everything that I'm going to talk about today should be found on our website, Office of Research Infrastructure, and I recommend that as a really good resource to delve into and find out information about the services and support that researchers across La Trobe have. The purpose of this session today is really to inform particularly new starters at La Trobe of the services that we have. We have a lot of services, but a lot of people aren't aware of them. And so this is a really good opportunity to become aware of what we have. And for those people that have been here for a while, for them to also refresh their memories on what we do have and hear the new things that we've made available. So I'm going to begin about talking about our research platforms. So what are our research platforms? Basically, these bring capabilities, expertise, and equipment together under a defined governance and management structure. So each of our platforms have academic leaders and a steering committee. The aim is to provide broad benefit across all research groups across the whole university. We want to make sure that we've got properly maintained equipment and facilities, but we also want to make a statement external to the university around the capabilities that we have and the feasibility of being able to do research projects for externally funded organisations. One of the most important things about our research platforms is the dedicated staff that we've got. And I'm really privileged to be working with a team of experts across different disciplines who understand and have extensive knowledge of their different respective areas. And you'll hear from a group of these colleagues today. And I think they're the people that really make a difference to our research and add value to the research outputs that we create. I'm going to talk, actually I'm going to hand over to Chad to talk very briefly about iLab, which is our equipment and booking and billing system. The system here is a system that you'll interact with, particularly when you're booking equipment through some of the equipment-based platforms, as well as statistics. So Chad will just give you an overview of it and then how to access that system. So over to you, Chad.
Thanks, Christopher. So yes, Christopher said iLab is our booking and billing system. It's known as a PQMS, a platform quality management system. And you can think of it as like a job desk where you would log jobs, log requests, that platform staff can then reply to in action, and you will also book equipment through that as well that you've been trained on. So you can find the very simply by going to Google and Googling La Trobe iLab, which should come up with our page. Up the top, you'll notice there is a login button. So because this system is integrated into La Trobe's login system, you can use your credentials for logging in. So when you click the login button, Christopher, if you could go forward. You'll get a few options. Now up the top, the red boundary box is moved, but you'll see line to sign in using Latrobe credentials. Now, if you are outside of Latrobe, an external user, you can use the Australian Access Federation credentials using your own university, but the majority of you will use the Latrobe credentials, which will take you to number 3. So you sign in as you normally would with your email address and password. Then it will take you to another screen. If this is the first time, you'll need to select your lab. Now this will be your supervisor, and typically it's the person who is in charge of your funds, the WBS funds. That person, they need to approve you into that lab and then assign you a WBS. If it's kind of sitting there, if you've logged in and registered and requested access, you may need to poke your supervisor to action that. And once they have done that, that will give you access to iLab. Now, in number 5, you can then access what we call the hamburger menu, that icon at the very, very top, which kind of looks like a hamburger, which will drop that menu down. And then from the My Cause button, you can then choose between the platforms which are on iLab. So bioimaging, Centre for Materials, Surface Science, Genomics platform. The Trobe equipment is our general equipment platform. Proteomics and statistics, who will be speaking today.
It's worth adding to that if people are looking at hosting equipment that they want to share and have a calendar for, we can set that up on the Latrobe equipment core here, and we can also collect funding if people are having cost recovery mechanisms for those bits of equipment. So that's something to keep in mind.
Thanks, Christopher. If you have any problems with iLab logging in, you can contact me directly on c.johnson@latrobe.edu.au.
All right. Now I'm not sure how to get rid of these screen.
It'll just look like a scribble board when you're done.
Yeah, that's right. Well, I'll let Chad keep talking while I work out how to get rid of those annotations.
Sure. So my name is Chad Johnson. I'm the optical microscopist in the bioimaging platform, which is a platform facility encompassing all of Latrobe's high-end optical electron microscopy, flow cytometry, histology, and analysis equipment. Excellent. So we are a centralized facility located in LIMS 2. So we have access to optical electron microscopy, flow cytometry, histology, and image analysis resources. Now we have a individual staff member who manages each of those capabilities, which allows us to have that expertise to assist you in any of those particular facilities. Next, Christopher. So just like a lot of the platforms that are going to be discussing, talking about their facilities today, we're involved in basically all parts of the of research projects, right from the very, very start when you come up with an idea that you may want to investigate, right through to experimental design, preparing samples, acquiring data, analyzing that data, visualizing that data, and presenting that data. Now we can analyze or image small objects right from down below 90 nanometers to sub-nanometer objects, right through to large objects or organisms, right up to macroscopic objects. So you can think of this as a scientific photography facility. Now we offer training and access to high-end analysis software such as Amaris, Zen, Flojo, FACSDiva, and Site Expert, and we have a full histology suite. So we have a full complement of equipment. We also have specialist staff who can actually train you to do all of the processing right from the start. But also if you're short on time, they can also process that for you. And we also offer script writing for automation of an image analysis. So firstly, optical microscopy. Now, I can talk about all of the equipment on the next page for hours. but I think it's just easiest to show you what we can actually output. So we have a full range of confocals, automated slide scanners, stereoscopes, laser dissection systems, but the proof is in the pudding if you see the next slide. So we can pump out amazing images. We can assist you with processing of your samples. Now we can image both fixed and live cell data. Oh, is it flicking forward? So we can produce multiplexed imaging, so multi-channels, three-dimensional data. So the brain section on the bottom was actually made up of 200 separate images, so tiled scans for large objects. A lot of these images, so we run an annual imaging competition. So a couple of these images were part of that. There was a couple of winners, and so we can put out some amazing fluorescent images, as well as stained chromophores, so H&Es. We have an automated slide scanner to be able to essentially scan 100 slides. So you can put 100 slides into the system, push a button, and then walk away, and then come back, and you've got your data there. So we have a full optical suite in the bottom of LIMS 2. Next slide, Christopher. So electron microscopy. So Dr. Boyan Liu is the manager of that facility. Next slide. So, of course, we have TEM SEMs. Now we can process. We deal with both biological and material sciences. So we deal with a lot of material sciences for those in engineering. And also industry. Now, a lot of people think that TEM is often just a 2D image, or at best we have SEMs which can get a topographical image. But we can also do three-dimensional imaging using serial block facing, which is essentially like having the word has disappeared from my mind. Oh crap. A sectioning system, which can section off the tops of samples and slowly make their way down, taking images as they go to produce three-dimensional images. For material sciences, problems with videos. For material sciences, we can not get just structural information, but also elemental mapping using EDS. So that uses X-rays to actually analyze the elements that make up particular substances. So we can get an actual quantification of the amount of the elemental makeup. We've got, of course, TEM, SEM imaging. We could do negative stain, cryo-TEM. So cryo-TEM and techniques such as TEM topography and TEM diffraction allow us to get very, very high resolution images. TEM diffraction has been used to get almost atomic resolution. So we can create structural information of proteins and biological materials. So flow cytometry, we have a full flow cytometry suite that is run by Dr. Bo Shi. Now we have multiple sorters and analyzers with different lasers that can image throughout the spectra. So we also have the biosorter, which can actually be used to sort large objects. Now, when we talk about large objects, these aren't cells, these are actual embryos. So zebrafish, Drosophila embryos, so we can actually separate those based on their size, their fluorescence. So all of that equipment is run by Bo, which is up on LIMS 2 level 4. The histology suite is headed by Dr. Jackie Orion and is staffed by Dr. Sarah Wilson, our EM and histology technician. Histology services allow us to use to prepare your samples, embed them in paraffin blocks, process them, section them, stain them. We've got a cryotome for cryosectioning. And we've also got the expertise to actually train you how to do all of this work from the get-go. So if you're going to be spending several years preparing tissues, preparing samples, then often it is just easiest if we train you to do it yourself. So we have a full suite of histological equipment, so cryotomes, tissue processors, and embedders. We've also got a cold plate, which can keep your sections cold so that the paraffin doesn't melt at room temperature. Now, once you've got all of this data, what do you do with it? You need to analyze it or you need to visualize it. So we have access to high-end workstations with commercial software as well as free software. We have Flojo licenses which we can distribute for personal use, so you can purchase them directly through us. We buy them in bulk and pass those on to you. We also can automate your analysis using script writing in Fiji, and we also offer workshops throughout the year for Fiji, ImageJ, QPath, Amaris, and there's also Flojo workshops throughout the year to allow us to impart that knowledge to allow you to do that work for yourself. So you can contact us through bioimaging@latrobe.edu.au. We're also on Twitter, Blue Sky, and YouTube. We post videos and updates through those. So thank you very much.
Thanks, Chad. Now I'm going to hand over to Rowan.
Hello, everyone. My name's Rowan Lowe, and I work at the Proteomics and Metabolomics Research Platform, which I'm here to talk to you about. So let's get started with the team. We cover, obviously, proteomics, metabolomics, and small molecule research. And that is, we've got me, Rowan, working in proteomics along with Bushra. We've got He and Jing looking after our metabolomics capability. And we've got Daniela, who's running the NMR small molecule structural work for the chemists primarily. And we've also got our esteemed director, David Greening. So what do? We are an analytical capabilities, instrumentation, expertise, and training, working on those. So these are a range of analytical methods covering, as I mentioned already, the three big ones, but we also cover some molecular interactions. We have vehicle instrumentation. We cover data analysis through bioinformatics. And yeah, we can train people as well to learn how to use all of these techniques. Next slide, please. So where are we? We're on the Bandura campus. We have two sites, one in the LIMS 1 building and one over at AgriBio. And that covers where all our instrumentation is held. And yeah, we've also over at AgriBio there's an independent lab, Baron Bowden's Spatial Omics lab, which I'll talk about a little bit later, but just keep in mind we're also tightly integrated with them. So proteomics, let's go through each main capability. Proteomics, we can basically do anything to do with quantifying and characterizing proteins at the molecular level. So ID, for deep proteome profiling, you want thousands of proteins identified and quantified. We can do that label-free or tagged using our LCMS instruments. We can handle a diverse range of inputs. Basically, we can tailor our methods to suit what you need to analyze. But typically, these are tissues, organelles or subcellular organelles like EVs, fluids like plasma, or it could be molecules. People are doing quality assurance on a recombinant protein, maybe a new protein drug antibody or something like that. We can do PTMs, which people are often interested in things like signaling in biology. So phosphoproteomics is a good one for tracking rapid signaling things. Also glycoproteomics for changing protein characteristics. And also similar workflows involve intact mass analysis of proteins. I've already mentioned BioCore for protein-protein or protein-drug interactions. And of course, we have a range of computers with specialist proteomics software to help you analyze your data. And we love to collaborate and come up with new methods tailored for people. Next slide, please. In the metabolomics capability, we've got T and Jing running this. So again, we're now talking about metabolites, similar technologies with LCMS and GCMS. And also we're introducing nuclear magnetic resonance into the metabolomics capability. So those technologies can measure, quantify, characterize metabolites in a huge range of sample types. We can do untargeted and targeted metabolomics, depending on whether you know exactly what you're trying to track or whether you're looking more for discovery of new biomarkers. And yeah, we also like to highlight we've got different sources of these metabolites, like volatilomics, which is just almost like what do things smell like? Why do things smell the way they do? Lipidomics is a great one for, again, cellular signaling. And yeah, we handle all of the data analysis and can train people on data analysis for their metabolomics data as well. And yeah, everything in metabolomics is even more bespoke and customized. So small molecule characterizations with Danny. We've got 2 NMR in the LIMS 1 building and access to the 700 instrument at AgriBio. And Danny does all of the standard stuff you expect for structural analysis of small molecules, but would also like to highlight real-time monitoring of molecules in the NMR for kinetics, binding studies. Drug protein binding can be done in an NMR instrument. And yeah, Danny is also introducing metabolomics workflows now as well with NMR, which is really cool. So great for any multi-omics there. Next slide. Yeah, and I also wanted to shout out Baron Boughton, and he's got his independent lab, which covers spatial multi-omics, but is very much aligned with what we do at the PMP. So I wanted to highlight that you can contact him if you're really interested in spatial omics. He's got a timsTOF Flex, which is ideally set up to detect a range of analytes in a spatial context. Next slide, please. So everyone always asks us, how much does it cost? Well, it's actually pretty reasonable. Some of the numbers can look quite big, but when you consider the amount of staff time we can save you and the costs of equipment, it's pretty economical at the end of the day. And it covers basically just staff time and consumables and a bit of cost recovery on instruments. We're not making any profits off academics. We give academics the cheapest rates, but we're pretty flexible, a range of options for everybody. And always like to highlight some of our services are totally free, so don't be afraid to ask. Next slide. So how do you get involved with us? How do you interact with us, get information? So we have an email list. You can get in contact with us. We can add you manually, or you can use the subscribe link. We have a quarterly email newsletter called Peak Perspectives. We've got a small mass spec users group if you're really keen to just nerd out and chat mass spec and meet some other mass spec users. You can catch up with us there. We have free training sessions throughout the year. What else? We've got a website. We're on iLab, as mentioned earlier. Please check out iLab and get registered, even if you're not really thinking about using it immediately. And I also want to mention we're even on YouTube. And please watch our YouTube video, pump up our numbers so that we get more views than genomics and bioimaging. It's really close between the three of us, so I want to see more views on PMP. And I think that is it, Christopher.
All right. Thank you very much, Rod. Now I'm going to hand over to Alicia to talk about statistics.
Hi, thanks, Christopher. So good morning, everyone. It's great to see such a big turnout. So my name is Alicia, and I'm going to introduce the statistics consultancy platform. Next one. Yeah. So the statistics consultancy platform work with both internal and external clients. So this include industry, businesses, government, academic researchers, and research students across all disciplines. We provide statistical advice, and I'll go through what exactly we provide shortly. So if you're planning to start a project that involves data, it's best to contact us before the project begins so we can provide advice on the statistics design and also the best statistical practices. However, having said that, our platform is very experienced at handling short-term queries as well. So feel free to contact us. So the next one, Christopher, thank you. Yeah, so the services we provide include guidance on study designs, including surveys, and advice on best approaches to data collection, so aligned with your research questions and the objectives, statistical support for grant applications, and statistical analysis of the data. And we also help with summarizing, interpreting, communicating, and writing statistical parts of you, for example, for your reports and manuscripts. And we also provide training in different areas of statistics and use of statistical software such as R, SPSS, and STATA. And these could be at introductory, intermediate, or advanced level. Next one, Stoffer. So here are a couple of free services available to La Trobe staff and students. So we provide 5 free hours of consultancy for external grant applications led by La Trobe staff. And we also provide a total of 5 free hours of consultancy to La Trobe research students. So I really encourage you to use these, and it's free, and also you can get to know us. So we have a number of affiliated statisticians. All of our staff have strong statistical academic backgrounds and well over 15 years of experience working as statisticians. And most of us are actively engaged with the Statistical Society of Australia. And I would also like to highlight that our team includes both senior statistical consultants as well as senior academic statisticians. So whether you're interested in statistics consultancy or a long-term statistical collaborations, we are the right place to reach out to. So across our team, we have strong statistical capabilities. So these include, among others, meta-analysis, biostatistical methods, analysis of data from clinical trials, forecasting, predictive modeling, big data, and so on. So regarding pricing, so we don't have any equipment or anything, so it's just the staff cost. And I've already talked about the 5 free hours of consultancy. And for La Trobe staff, the cost of accessing the platform is reasonably low. So it's only $100 per hour, highly subsidized by the university. For external non-La Trobe affiliated staff, clients are charged at $250 per hour. And previously, we've been quite flexible. So, for example, you can contact us for the possibility of working with a statistician at a pre-specified FTE. And also, you can contact us if you are interested in advertising a short-term unpaid internship to our Master of Data Science students here at La Trobe. Yeah, so the best way to reach us is via email or via filling out the inquiry form via iLab, as Christopher and Chad mentioned previously. I think that's it from me, Christopher.
Tanmay, I'm going to hand over to you.
Thank you, Christopher. I'm Tanmay Shekhar. I'm the Senior Manager for the La Trobe Animal Research and Teaching Facility. In short, stands for LOFT. So I'll be referring to it as LOFT moving forward. So it's a platform to support animal research here at La Trobe, and we do that by providing state-of-the-art facilities for holding and housing animals, a variety of species, and providing services to support compliant and high-quality animal research. And as you can see in this map, we are mainly based at the Bandura campus, and we are spread across multiple locations within the campus. So we've got the main central animal house within the animal glass houses compound, which is complemented by a few annexes building. We do have a small animal house facility in AgriBio. We do have animal facilities within AgriReserve, Zoo Reserve, and across some of the biological sciences, the limbs and breed building. I'll go through each of these a bit more in detail so you understand the purposes of these facilities. So the AgriBio Small Animal House and the Central Animal House SPF. So these are our high barrier facilities. So the primary purpose of having multiple facilities, but also having measures to prevent contamination of the facilities is to safeguard the health status of the animals that are used for research, just to control all the variables. So this is where we have the small animal house and SPF. It's considered the highest barrier within our suite of facilities. And these facilities are equipped to hold mice and rats primarily for breeding and experiments. And these are all BC2 certified facility. Our AgriBio facility is primarily a breeding facility where we have multiple genetically modified or native, not native, but wild type mouse colonies. And that just allows us additional biosecurity measures to safeguard our precious GM colonies. And SPF, on the other hand, has been designed primarily for experimental animals requiring high health status. Next one, Christopher. Thank you. Within the central animal house, we have conventional area, and this is the area that is primarily used by researchers broadly for a range of their experimental setups. So animals that are bred in a small animal house, or you could procure animal from other facilities, they come into the conventional area. And this area has been designed specifically split into multiple zones to cater for a variety of animal research. So we've got the short-term holding areas for mice and rats. We've got dedicated mouse behavioral suites and rat behavioral suites. In addition to that, within these areas, we've got multiple capabilities such as dedicated surgical suites. We've got equipment such as ultrasound machines, a radiator, electrophysiology rig. And with the behavioral suite, it's a variety of behavioral equipments available to support the behavioral research here. Next one, please. Within the conventional facility, we have also have a aquatic facility, which is the PC2 aquatic certified facility. It's used for housing our zebrafish. The zebrafish research has been growing rapidly over the past few years. So only just a couple of months ago, we did a recent expansion where we went from 250 tank capacity to 450 tank, which has been really good. So this is primarily holding zebrafish, GM, non-GM again, and it's equipped with micro-injectors for creating CRISPR strains, and that's complemented with some microscopes to do some preliminary analysis on that. We do also have a quarantine room available in case we are importing fish embryos from other facilities, thereby safeguarding the health status of our main facility. Next one, please. So we've also added DanuVision, which is a zebrafish larvae activity monitoring instrument. It enables for behavioral tracking, and it's used quite regularly in drug development using zebrafish larvae and embryos. So this was recently added, and it's of a great use. I would encourage people to start using that. Next one, please. That leads me to the other areas. So those were the primary areas. So the other areas, what we call it as external areas. So these are the areas that were designed and developed primarily from catering other species. So whatever species I spoke about, the laboratory rodents and zebrafish, they're very tightly controlled from the health status, but we do have a lot of ecology and zoology research happening at La Trobe as well. So these facilities are designed for that. So I won't go too much into details, but historically we've had wide variety of species. That just shows the breadth of variety of species we can hold, but also from the services that our team can provide to care for these animals. So how do we support research? So apart, like I highlighted some of our key facilities, apart from providing very project and species appropriate facilities, we do have a great team of technicians that provide high level of animal husbandry and supporting activities to keep to safeguard the animal welfare, but also make sure that your research outputs are not compromised. So a couple of key things that we do is establishing and maintenance of the breeding colonies. And now this is something that we really take pride on, is we do manage over 90 different mouse strains and equally about 90 different zebrafish strains as well. And so we got a team that take care of day-to-day maintenance, but also monitor the breeding colony performance. They advise on advice on maintaining the strain integrity, but also forecast and make sure that you have the animals available for your experiments at required times. In addition to that, to support the experimental research over here, our team of technicians are quite skilled in a variety of procedures that are associated with the animals, and they are available to assist for routine administration to health monitoring or any other project-specific requirements as well. Next one, please, Christopher. Yeah. And a core of that is also that animal research can be a bit complex, navigating through complex regulations and requirements and maintaining compliances. So we have a team of vets, and we provide preliminary consultancy. Like we said, as soon as you think about doing animal research, it's best to contact us so we can guide you through animal ethics application, also from your animal research project planning purposes, what works practically, what doesn't work. So it's always good to get those bearings right to begin with, and that just allows you to have more successful outcomes in terms of ethics approval, but also ensuring that you understand the constraints for the animal research. Training and compliance is a big part in animal research, and that's why we've got teaching in our platform name as well. And so we do provide training to all facility users that are coming in. And training is a requirement by the code that only competent people can perform animal procedures. So that's why it becomes a big part. We do offer free training for Latrobe students and researchers for the common techniques, and that's part of the services that we do. And all of this data from animal management, animal records that needs to be kept and needs to be centrally kept. So we do have Ticket Lab, which is a cloud-based animal management database. So it ensures compliance with your animal records, and also makes easier for reporting purposes. Apart from training, everything else is charged for the platform. So the charge rates are available on the intranet for the LTU users. Thought I'll leave you with a bit of an example of how we support animal research. Recently, there was a publication by Trobe and Grant Robbins group where they used mouse models to assess whether the amnion epithelial cells could be used as a therapy to inhibit hypertension-related vascular pathology and cognitive impairment. So and they had a publication in Nature Scientific Report, which was quite a high impact. And the way LAF supported their study was their colony was established at AgriBio Small Animal House. Then in consultation with the research group, we projected their experimental requirements so we could set up and manage the breeding that way. So when the animals were ready for the experiments using in-house transportation, we transferred the animals to the central animal house where they were housed for experimental procedures and specialized surgeries. While this was going on, the LAF training team and the vets provided training and competency assessment to the researchers and students so they are ready for the procedures. And while the experiments were going, the LAF team also, apart from performing routine husbandry and checks as per the experimental requirement, we assisted them in any project-specific health monitoring, substance administration, and then LAF vets as well provided assistance with surgical mice and any of unforeseen complications that would arise. So this is how it all ties in together. It's complex, and it's supported by a team of about 16 to 18 staff members. And you can contact us by looking at the details at the website mentioned here, but also on intranet. But usually the first point is to use our the platform email address, laft@latrobe.edu.au, and we can take your queries and direct it appropriately from there. Thank you.
Great. Thanks very much, Tanmay. I'm going to hand over to Lauren now.
Thank you. So hi, everyone. I'm Lauren. I'm the Senior Manager of the Clinical Trials Platform, and I'm going to tell you about our support services that we offer. So our team, who are highly experienced, have provided researchers with training, expert advice and services for the support and conduct of clinical trials, and to the highest possible standards. So our broad capabilities include advice on trial design and methodology, grant development, ethics and governance submissions, database design and build, data management, support for clinical trial setup, participant recruitment, and support with public and consumer involvement. So just go into a little bit more detail about what that looks like. So I guess we can provide support at any stage of the trial life cycle. Obviously, the earlier the better to get us involved. If you've got a concept or a grant that you're applying for, that's really the best stage for us to be involved. We can help with that concept development, refining your proposal, refining the grant application itself, and reviewing that. Help with budgeting and costing to make sure that any platform services are budgeted for right at the start and costed appropriately. We can help with protocol writing, sample size calculation in consultation with the statistics platform. We can help with health economic impact analysis, support public and consumer involvement, help with identifying trial sites, and contract facilitation. Next slide, there we go. Oops. Jumpy. And then ethics and governance support. So our team can provide advice on the ethics plan. So HREC and governance submissions, we can actually do that work as well. Some of our researchers will contract that out to us because it's just more efficient. It allows the study to get started a lot faster and prevents sort of delays back and forwards between ethics and governance officers in our team. We can help there is response to queries, indemnity and insurance advice. We can help with standard operating procedures, so support with setting those up for your trials. Data safety and monitoring boards, so that any data coming out of the study is reviewed and compiled independently and prepared for that data safety monitoring board to review. Adverse events, we can help with the monitoring and reporting of those. And then annual and breach reporting and corrective and preventative action implementation. Next slide. Thanks, Christopher. And then the data side of things. So data management, database build, that's quite a lot of what we see contracted out to us as well. So either using REDCap or Question Pro, those databases allow for things like e-consent, participant surveys sent directly to participants for them to fill out in real time, and sort of allowing data collection from all people. Independent randomization service as well. We can offer that and build that either into the database or as a separate standalone database. We can work on helping with your data queries, data extract, linkage, and integration with other databases. We can help you with source data document templates. If you're not using a database for all of your data, source data, we can provide advice on data management and storage and data sharing as well. Another capability is monitoring. So our team can provide desktop or remote monitoring as required, and risk-based monitoring as well, and developing monitoring plans. And we can help prepare for audits and regulatory inspections as well. And then more on the data side, so cleaning and analysis sort of at the end of the study, we can help with the statistical analysis of the data in consultation with the stats platform, endpoint adjudication, independent safety monitoring, and those data safety monitoring board reports that I mentioned previously. We also provide some free education resources and training. So we can help with protocol tools and templates. If you're getting started, you're not sure what you need to do in terms of a clinical trial protocol, we can help with templates for that. Other template documents you might need, like a abbreviated CV to show all of your expertise and training and experience, and delegation and training logs so that you can show who's going to do what in the trial and who's overseeing that and what their training is in relation to that task that you're delegating. We can also help with investigator site file templates. So if you're setting up the trial, we can help with getting that filing system sorted so that all of your sites are using the same filing system and everything's filed appropriately. One of the trainings we offer is a trials methodology seminar series. And so we have a range of topics where presenters speak on those, and we have those sessions recorded. So we have a bit of a back catalogue of those available on our website. And we have another session coming up in a few weeks that I'll tell you about as well. We also offer good clinical practice training. So that GCP training is required if you're doing any research on humans the Research Ethics Office will ask for that training certificate. If you do that training, that certificate will last for 3 years. And it's a good way to sort of get a good understanding of clinical trials and the whole research life cycle and what you need to do and what you should be doing. And we also provide information on upcoming conferences and events as well. So these are our training events that are upcoming. So good clinical practice training, we have sessions in May, July, September, and November this year. And we offer both introduction and refresher. So I mentioned that certificate lasts for 3 years. So if you've already done prior GCP training, you can attend one of our refresher courses. Or if you're new to clinical trials and to research, we offer the introduction sessions as well. And that's asynchronous learning via the LMS. And so you do some pre-reading and then attend a live session where we cover off things that might need to be reinforced from the pre-reading. And then our trials methodology seminar series I mentioned, we have an event on the 29th of April. This one's titled Beyond Medicines, Adverse Event Reporting in Non-Drug Trials. So you can follow us on Eventbrite to keep up to date with our events. And finally, just to cover off all of our, I guess, capabilities, we can also offer complete project management as well. So some of our researchers use this service, and it can be just basically anything they need support with to run the project. So again, it's setting up a review of the site files, creating investigator training packages. So if you need to train people at other sites, we can help with that training as well. Organise and facilitate that investigator training, support for the inclusion of culturally and linguistically diverse communities. We can help with advertising and promoting trials, trial registration through bodies like ANZ CTR, monitoring participant recruitment and protocol compliance. We can also organise and host events, so focus and consultation groups, roundtables, facilitate and organise consumer group activities as well. So if you wanted to maintain independence from that group, we can sort of organise and run all of those events and collate feedback from consumers to shape your projects. And then at the end of the study, once that's all done, archiving as well. So just a few examples of some researchers we've helped to date and some specific services we've provided. So cell therapy for acute stroke. This is a project where we've provided some project coordination, helped with some site selection, developing a risk management strategy, vendor coordination and management. We did some public involvement focus groups for that, and we're working on HREC amendments for that one as well. Another project, suicide prevention in farmers. So with that one, we've done HREC submissions, protocol amendments, performed a database build that incorporated e-consent, automated surveys to participants, and some clinician-entered information as well. Also included randomization in that database as well. We helped with investigator site file setup, developed safety reporting guidelines, and did some project management on that project where we did the group allocation. So we did the matching of peer workers to participants and managed the control group as well so that the team was able to stay independent and to stay blinded. And so how to contact us. So email is the best way, ctp@latrobe.edu.au. That's our team there. So led by our academic director, Joe Kemp, myself, the senior manager, Mira Center, and our project coordinator, and Rashmi Bhatt, our database specialist. And you can follow us on LinkedIn as well to keep up to date on what we're doing. I think that's it. Thank you.
Thanks, Lauren. I'm going to hand over to Ishmael.
Good morning, everyone. I'm Ishmael, senior bioinformatician at Genomics Platform. Yeah. In Genomics Platform, we provide a range of the cutting-edge sequencing equipment, such as 10x for single-cell, nanopore long-read sequencing, as well as Top Station for the quality checking of the DNA and RNA. Next slide, please. In the sequencing services, we provide both short read and long read sequencing opportunities. which organized by my colleague Asha, and we provide library preparation, sequencing, both Oxford and the library preparation for the short-read sequencing, as well as single sequencing, plasmid, and 16S sequencing for microbiome analysis, as well as contribution in the grant support, grant application, providing cost estimate for the grants, and anything which can help you to be more competitive in the grant application. Next slide, please. In the bioinformatics services, we provide a variety of complex bioinformatics and machine learning services, such as transcriptomics, RNA-seq, small RNA-seq, and alternative splicing analysis, metabolomics data analysis, post-network and enrichment analysis, de novo assembly, variant discovery, single-cell analysis, which allows you to get the transcriptomics picture of this transcriptomics picture in the cell level, microbiome, and recently virome data analysis, meta-analysis, as well as integration of the different source of the data, which we call that multi-omics and data integration. Also, we provide data analysis services in the epigenomics level, both chip-seq methylation, and more importantly, we provide a range of the machine learning services such as random forest, deep learning, association rule mining, which are very important when you are working with a huge amount of the data, or you are getting the data from the public resources you want to analyze and get a pattern out of them. Finally, we provide the training in bioinformatics and machine learning. We found that, one by one or individual training of the postgraduate student is more practical because the student can bring its own data, and we help her to find the best way of the analysis of the data and get it published and so on. Next slide, please. One of the area which we put a lot of efforts in the last couple of the years is setting of the virtual machines for researchers and students. To give you a brief overview, in Latrobe we have two separate resource of the big data analysis. One of them is the HHPC, which is very big computer handled by the IT Unix team. And in addition to that, we have the grants from the Nectar and previously from the Oracle, that in those platforms, we generate individual virtual machine for the researcher, and the researcher can have, using the simple laptop, can have access to the big virtual machine on the air. It completely belongs to them. For a defined period of time, they can run the analysis on the machine, they can install the software themselves, or we can show them how to install that. And at the end, it had a lot of flexibility. They do not need to wait for the queue for the resources. It's more flexible and good for the training. And also, we help the student in the preparing the manuscript in the bioinformatics machine learning, as well as the contribution in the grant application at CI or AI. And one of the examples is that, which has been published in Nature Plant, is providing a catalog of the seed germination in the single cell level, which is really impressive. And you can see that publication here. Next slide, please. And how you can contact us. The best way in using the genomics Latrobe email, and our team includes Professor Matt Lucy, our academic director, and Asha, which is our experienced lab technician working with both short read and long read sequencing data. In the bioinformatics, you can contact me or manager for any help. And the most important thing is that we are available to help the student from the beginning on planning of the experiment, understanding of the data, performing the sequencing, analysis of the data, and final publication. Feel free to reach out if we can, if you have any question regarding bioinformatics, sequencing, and machine learning. Thank you.
Thanks very much, Eshma. I'm going to hand over to Nick.
Okay, thank you. Hi, my name's Nick May. I'm the Manager of Digital Research. And I basically, I can be quick to get us out of the door on time, but I'll concentrate on the important things for your research and how digital research relates to that. So what is it? Well, it's really, you've seen some great instruments and specialist services, but we cover the general digital research aspects that support your research. You're actually doing your research. So from data collection, processing, visualization, things like that, data publishing. So yeah, in the research lifecycle, and we characterize that mainly as technology, data, and skills. So the servers, the software and tools, the data storage, so data publishing platform, databases, and the skills. We have the training program that we run throughout the year and irregular research events. So for instance, this Friday, we have a webinar run by the vendors of our online research notebook. So I'll tell you how to find out about that link if you're interested in attending that webinar on Friday. So we provide a suite of digital resources that are documented on our website, which we call a standard offering. This includes those compute, software, storage, etc. And that's available to all researchers and all research projects. And we also importantly provide information on how to request additional resources if beyond the standard offering. Okay, so the team, we're a small team. We mainly work through collaboration with the library and information services and with external providers. We provide advice, training, and we looked at how we can improve our digital skills. So there's myself and our digital research analyst, Dr. Sam Ryan. Now, he's provided by an external partner called Intersector Australia, and that's important with how we collaborate externally. So in terms of support, as I said, we collaborate with the library and with information services, and we can connect you up with the appropriate people where needed. But we also have the Digital Research Analyst Network. Sam has his expertise, but if it's beyond his or he needs extra help, he can consult a network of analysts through Intersector Australia who are embedded in universities across Australia. So it provides that wider technical and expertise in how to do digital research that can support you. So they can discuss this technology data needs, and we can connect you with the appropriate services. So finally, just how to contact us. We have the website. That's the best place to look to start off with on research infrastructure, digital research. You can email us at our email, and you can subscribe to our mailing list. You just send a blank email. I'll put it in chat. A blank email to that website address, and you'll get the digital research news, which comes out occasionally when we have some news. So for instance, this afternoon, I'll send out a reminder for the online research notebook webinar on Friday. So yeah, just send a blank email to that and you can contact us through those. But I would always start looking. There's a lot more detail under each of those categories on the website, and you may find the answer there first. Okay.
Great. Thank you very much, Nick, and thank you to all of our speakers today. Again, just want to remind you of our website. We have the Office of Research Infrastructure website, so click on that and have a browse at that and get in touch. All the contact details are on there as well. Again, I'd like to thank all our speakers and encourage you to access our facilities.